Okay, that was one dimension, but it scales up to 3 dimensions. And I can write down like, the concentration as a function of time, derivative of species I should say, is equal to the diffusivity of species I times in 3D space del squared times Ci, and then plus reaction terms, etc. Now, if we want to take this equation and we want to solve it, we need a couple more things. We need initial conditions and we need boundary conditions. And you guys all know what initial conditions are probably, so I'm going to talk about boundary conditions here. We need boundary conditions. So as we talk about boundary conditions in the next few minutes before class wraps up, we're going to assume that our system is one-dimensional again and at steady state, right? So then our equation is 0 on the left-hand side equals to our diffusivity times d squared c dx squared plus reaction terms, et cetera, which I'm not going to write out explicitly at the moment. So what kind of boundary conditions are we likely to see if we're going to model spatial systems in biology? Okay, so let's pretend like we have a system here. And I apologize for this. In the next few examples, I'm going to be flipping back and forth between the x-axis going vertically and horizontally. I don't know why I did that. But we're going to pretend like we have, x going vertically, and it's 0 at one boundary and x equals to L at the other boundary. Pretend like what's going on here is that we have some reaction terms where at some point, and that little empty set is supposed to denote like nothing. So from nothing comes this molecule, which is synthesized. Basically, that's a synthesis term. And the molecule could also be degraded. So it could also go back to nothing, at least nothing that we're modeling. And you could have diffusion, which is usually represented by like a jagged molecule, or sorry, jagged arrow that looks like maybe the molecule's like bouncing around randomly, right? So the first set of boundary conditions and the simplest kind of boundary conditions you can encounter is what are known as no flux boundary conditions. And if no flux boundary conditions, basically you have a molecule, and if it hits the boundary of your system, it basically bounces off. Right? And that could happen on either side. That's called no flux. Molecules can't flow out of the system through the boundary. Right? It just bounces right off. Now to model that, recall that this diffusive flux, J, is equal to the negative times the diffusivity times dc dx. And if this diffusive flux is 0, that means dc dx evaluated at the boundary is equal to 0. And in our particular case, the boundary is located at x equals 0 and x equals L. So dc dx at 0 equals dc dx at L equals 0. And sometimes you just see this simplified. Instead of showing both of these, sometimes it's dc dx evaluated at 0 comma L equals 0. Just another way to shorthand write it. Now, there could be some cases in which instead of no flux, let's assume that we have a system. Now I'm going to have X go horizontally. Let's say that we have a system where we have one boundary on the left-hand side, another boundary on the right-hand side. Left-hand side is X equals 0. Right-hand side is X equals L. Let's say that for whatever reason, in some way, the left-hand side is constantly producing our molecule of interest. This is what's known as constant flux at X equals 0. And of course, the molecule can still diffuse around with some diffusivity D. And let's say that you still have no flux on the other side. So if we have a constant flux, then our flux evaluated at X equals 0 equals to some constant. Let's just call that constant equal to Q. And so you have a boundary condition where negative times the diffusivity of dc dx evaluated at X equals 0, that's equal to Q. And then, like I said, the other side is dc dx evaluated at L equals 0, because that's no flux. Okay, so we need these ways to constrain our system so that we can actually do solve the differential equations. Last one that I want to go through today here. I got through 3 pages of 7 pages of notes. Last one I wanted to go through today is what happens if we have a system where there's a reaction taking place at one of the boundaries. And this is what's really important for biological systems, because a lot of times the boundary of your system is the cell membrane. And a lot of times there's reactions taking place specifically at the membrane. Okay, so imagine that we have a system where we have a cell membrane, which I'm going to draw here up top. And molecules generally can't get past the cell membrane. You have a receptor expressed on the cell membrane. And I guess I should write here that above the membrane is the extracellular. And below it is intracellular. This is another one of those cases where I have X going vertically, but now it's going to go down, where X equals 0 is at the top. Okay, so you have a receptor, and let's say this is an active receptor. So I'm going to put a little ligand inside of it. And this active receptor, because it's activated, will bind reversibly to some signal transduction molecule on the inside. So the signal transduction molecule could get bound to the receptor, right? And this is reversible. So it could be bound to the receptor, it could just dissociate from this receptor on the inside. The receptor is an enzyme, usually it's a kinase usually, this active receptor. And so with some reaction rate, say Kr, which I'm not going to write here, well, maybe I'll write it. So with some reaction rate Kr, you reproduce the original receptor without the signal transduction molecule bound to it, and then you release that signal transduction molecule, but now it's been phosphorylated P. Inside the cell, this signal transduction molecule can diffuse around with diffusivity D. I'm going to call this D sub A, because when we're modeling this, we're going to say that the signal transduction molecule without the phosphatase, without the phospho group added to it, that's species A. And when it does have the phospho group added to it, that's species B. Okay, so species B can also diffuse around inside the cell with diffusivity dB. Last thing to add here is that this phospho group could be removed and reproduce the original phospho group less signal transduction molecule, and that is done by an enzyme called phosphatase. And that has a reaction rate Kp. And that happens inside the bulk of the cell in this example, right? So you have a reaction taking place in the membrane where our signal transduction molecule A is reversibly binding to the active receptor. And then with some probability, which is represented by Kr, it can be phosphorylated and released from the receptor. Then inside the bulk of the cell, you can have phosphatase dephosphorylating this molecule. Okay, so let's write down some differential equations that describe that. We have 1 minute left. I did all this great setup on this example. Let's just write this down, okay? So, I think I'm just gonna have enough time to write down the bulk reactions. Okay, so this will be a good practice to think about the bulk reactions. So at steady state, we have diffusion of A times the second derivative of A in space. Sorry, diffusivity of A times the second derivative of A in space. And A is produced by the bulk reaction, which is the dephosphorylation of species B. So you have a reaction term here. It's a bulk reaction term. It's not happening just at the membrane. I can write down a differential equation for B. So that's a diffusivity of B times the second derivative of B in space. And it is consumed by the phosphatase reaction. Okay, so those are our two bulk reactions. And they include diffusion of this molecule inside the cell. So the cell is big enough, or the diffusivity is small enough, that because the reaction is taking place at the membrane, there is the phosphorylation reaction, there is a significant spatial gradient within the cell. Otherwise, why model diffusion if there's no spatial gradients, right? If diffusion's so fast, everything mixes inside the cell so fast, you just consider the cell a well-mixed system, kind of like a CSTR. And then you don't worry about diffusion. So we're just saying that the diffusion's slow enough that there's some sort of spatial gradient in the system. Now, what we need to do is we need to write down the boundary condition at the membrane, which is where the interesting reaction is taking place. So we can write down these boundary conditions in the following way. So at x equals 0, you have that boundary condition. And I'm not going to deal with a boundary condition that's far away from the membrane, just one boundary in this example problem. So at x equals 0, you have, at steady state, you have the negative d diffusion of A times dCA dx evaluated at x equals 0. That is your flux. That's your flux away from the membrane. And then you have the reaction at the membrane, plus a K on times the concentration of A evaluated at 0 times the receptor concentration, minus K off, because that's your other reaction associated with the molecule A, times the concentration of the Ca times receptor complex. So this is your equation governing your reaction at the boundary for the molecule A. For the molecule B, you also have a flux term. And B is formed by the forward reaction, the phosphorylation event, which is denoted by the rate constant is Kr for that reaction. Kr times the concentration of CaR complex. So another way to read this is that we're looking at this little shell of a boundary right next to the membrane. And in this little shell, which I can draw kind of slightly bigger than that, bigger than a little shell, but that's okay. It's a small enough distance that we're essentially saying this is all x equals 0 here. This is a super small like delta X width here. In that small little shell, molecules will arrive into that, at that surface, at the boundary, with some flux, right? So you have flux of A coming towards the boundary. And inside the boundary, you have this reaction happening. In the same way, molecule B is produced at the boundary, and you have its flux away from the boundary. And so that's how those things balance out. And of course, they have to balance out in a very particular way. The units have to match up, for example. So I said this is a flux here. The first term in both equations is a flux. Did these other terms, do they count as, in terms of units, they have the same units as the flux? So let's check those out. Let's check out the units. Okay, so flux, that would be the first term in both equations. Flux denoted by J has units of moles per area times time, which should have the same units as your diffusivity times dCA dx. And that's actually true. I think we did that last time, right? So the diffusivity times the first derivative spatial of concentration, has the same units as flux. That's why that is a flux. Okay. But let's take a look at that. So the diffusivity has units of area over time, or length squared per time. And then concentration has units of moles per volume, mole per vol. And then per dx has units of length. So this does eventually translate into the units of flux that we need. Okay, so that's flux, and we knew that was true from last time. Okay, what about these other two terms in the boundary? So K on times Ca times R. So we have a second order reaction. We have K on times Ca, which is evaluated at 0, x equals 0, times R. Now this K on is a second order rate constant. So it has units of 1 over time times concentration, or moles per volume. So that's K on. Concentration, of course, has units of moles per volume. So those two things semi-cancel out, and the combination of K on times Ca gives us what looks like to be a pseudo first order, right, time inverse, together. Right, so the first two factors combine together to give us inverse time. And the receptor, well, the receptor it's a concentration, right? But it's not a bulk concentration. If you notice, backing up to the boundary conditions themselves, the concentration of A is evaluated at x equals 0. But R, the concentration of R, it's not evaluated at x equals 0 because R is only at x equals 0. It's nowhere else in the problem. So you don't actually have to explicitly say that. It's always there at x equals 0. It's not anywhere else. Same thing for the AR complex. It's only at x equals 0. So R is only on the membrane. It's not a bulk concentration. This is a surface density concentration, a surface concentration. And so the units of R are in moles per area. Okay, so when you have a moles per area times an inverse time, then of course you have moles per area time. And that's a flux. So these units, they match. Similarly, the units of K off times the CAR complex concentration, they match up in the same way. So K off is a first order rate constant, so that's time inverse, and concentration of the CAR complex that's also in moles per area, because it's a surface density as well. So these things all match up, and so all the fluxes and the terms, they combine correctly, at least unit-wise. Okay, so the beginning of the next page, we actually, I have something there that I already said, but I'll go ahead and say it again because I think this is a slightly difficult concept. So why are these boundary conditions? You basically at the boundary here, we're doing a balance on a control volume near the surface, right? So you have the surface, you have the receptor on the surface, and the receptor is being bound by A in this sort of reversible fashion, right? So if we're doing a balance around this control volume near the surface, A has to first arrive at the control volume, and then it has this reversible reaction with the receptor. So the net flux in of A is balanced by the net consumed by reaction. So flux is a little bit weird because it's a vector, right? But on one dimension, it's either positive or negative, and so the direction of the flux is actually being telling you whether it's positive, is being told to you by whether it's positive or negative, right? So here, it's a little bit sloppy what I said here. I said net flux in of A is equal to net consumed by reaction. Now, consumption by reaction, that's not a vector, but flux is. But the flux into the control volume of A, this is, don't forget, X is pointing down in this problem, right? So the net flux into the boundary of A is actually the negative of the actual flux, right? Because flux we're adopting is positive when it's going in the positive X direction, right? So the net flux in of A is actually negative of J. So this guy here is D times dCA dt, sorry, dx, dCA dx, evaluated at 0. So usually you have a negative sign there. That's what Fick's law says. But the net flux in is positive D, and that's equal to the net consumed, which would be minus K on CA 0R plus K off. I'm running out of room, CAR complex. Okay, so that's why, so this is the balance makes sense when you think about it this way. And so that's why if I'm going to scroll up to the purple equations again, the flux term is on the right-hand side in those equations, but it has a negative, right? So this is the net flux out, and that's balanced by the net consumed by reaction to equal to 0 on the left-hand side. Okay, yeah, and you can write the same analysis for B, right? The net flux out of B is balanced by its production. Okay, so in all the equations that we just wrote down, what's missing? And what's missing is there's no equation for the concentration of CAR complex. I didn't write that down. I wrote down bulk equations for A and B and boundary conditions for A and B, but what about this CAR concentration? I didn't write that down. And so that's just another balance equation, right? So the equation for CAR is D of CAR dt, the accumulation, is equal to K on times Ca at 0 times R minus K off plus K reaction times CAR complex. Right? This is just looking like a receptor with endocytosis equation that we had before. The form is the same. It means something slightly different, but the form is the same. So you have this reversible on-off reaction, binding, debinding, and you have this forward reaction. So this is just even though this is only at the surface, this is just a regular balance equation. Accumulation equals net reaction. And there's no in and out for CAR. And similarly, you can write down an equation for R, which I don't actually have in my notes because it's just similar to what it was before, but I think R is only participating in minus K on Ca 0R plus K off. Actually, I think this also has a Kr term here. Because when CAR is reacted, you get the receptor back again. So these two things are negatives of each other. All right, so this would be a full description of this system. If you wanted to talk about diffusion of A and B in the bulk, and then reaction at the membrane. Actually, it's not the full description. I lied. So there's also another boundary condition, right, that I mentioned. There's a boundary condition at x equals L. So what should I write for that? So maybe L is far away. So far away that the phosphatase basically takes care of everything by the time it gets that far. And so then you have the limit of Ca as x goes to infinity equals 0. And the limit of Cb as x goes to infinity equals 0. So you have other boundary conditions that you could write down in this problem. Okay, actually, okay, I'm sorry, I keep making mistakes. Let me back up a little bit. This is not correct. This is the limit of Ca as x goes to infinity is some sort of Ca bulk. Right? Because as you get farther and farther away from the membrane, everything is dephosphorylated, right? So you have some bulk concentration way out there. Okay, so it's not going to 0. Cb does go to 0 because B is basically dephosphorylated so much that as you get far enough away, there's no more B anymore. Okay, so this is the kind of problem where there's a lot going on, right? You have diffusion, you have reaction at the membrane, you have reaction in bulk. You have to take into account membrane species with their own equations that you have to keep track of. So that's pretty complex. We can try to write down this kind of model in MATLAB and solve it. It's a little bit difficult because you've got to solve PDEs, right? We can convert this into a series of ODEs by discretizing space.